Hey, in this video we're going to be talking about the process of mitosis and meiosis. And it's probably going to be pretty quick, and I'm probably not going to know what I'm saying, but here we go. Okay, so mitosis is the act of cell division, or cloning, and it happens everywhere in the body, in like all different cells, and what it helps with is cell growth, maintenance, and it helps repair damage. The first phase of mitosis is called interphase, and it actually precedes mitosis. So I'm going to draw it for you now, and I'll come back when I'm done. Okay, so I probably shouldn't have written words because they're probably backwards, but I'll explain them anyway. So this is interphase, and here, like, all this spindly stuff, those are the chromatin, and they aren't formed yet. So they're just kind of moving around, you know? And this whole thing here is the nuclear membrane, and it's full and intact right now. This interphase is basically everything at rest. And then here we have the centrioles, and the stuff sticking out looks like a sun, kind of. Those are spindle fibers. And other phases, they're going to grow out. Okay, so now we're on to prophase. Okay, so here in prophase, the nuclear membrane, as you can see, is starting to kind of disappear and dissolve. And that the chromatin have just paired up and kind of straightened out more. And as you'll notice here, the centrioles have kind of split up and the spindle fibers are starting to grow. Next is metaphase. Okay, so now the chromatin has formed sister chromatids down the the equator of the cell, and the centrioles have fully moved over to the poles, and they're using their spindle fibers to reach towards the sister chromatids. Okay, so now we're on to a fun one, anaphase. Okay, so here in anaphase, the centromeres divide, and as you can see here, the chromatids have split up, and the individual chromosomes move to opposite poles. See, each of these lines here connected to the spindle fiber is part of those. Now we're on to the second last phase of mitosis, telophase. Okay, so here you can see it starts forming two new cells, and there's a whole new nuclear membrane forming on both sides. And as you can see, the centrioles have both their spindle fibers have really just started to dissolve, and they're getting small again, just like they were at the start at interphase. And the chromosomes, they're going back to chromatin, so they're going to be all spindly again and weird. And now here we are at the last step of mitosis, cytokinesis. As you can see here, the cells have fully split up and formed two whole new cells. And so it's basically back to where it was at interphase. Like, the centrioles are there again, the spindle fibers are small, the chromatin's just moving around in there, everything's fully formed, it's just two new daughter cells. And so that's the end of mitosis. Now we're on to phase 2 of the video, meiosis. Okay, so meiosis has two different rounds to it. There's meiosis 1 and meiosis 2. And we're going to start with meiosis 1. Okay, so meiosis 1 starts a lot like mitosis. There's interphase here, you know, the centrioles are just chilling out, spindle fibers are small, and the chromatin's going nuts. And then it moves on to prophase 1. And in prophase 1, it's pretty much the same as mitosis again. The spindle fibers start to grow, the nuclear membrane disintegrates, and, you know, these form. So it's pretty much the same there. But also, homologous chromosomes come together to form a tetrad, and this is a process called synapsis. And a synapsis is when two homologous chromosomes pair prior to segregation, which allows for possible crossing over. And crossing over is an exchange of genetic material between homologous chromosomes, and it occurs during synapsis. This increases genetic variability of sperm and eggs. And the chromosomes coming together and being so close together are what makes crossing over happen. And this forms entirely new gene combinations not seen in parents. So metaphase 1 is fairly similar to mitosis. We have the tetrads lining up on the equator of the metaphase plate. The centromeres align, and all the chromosomes lined up in the middle have one spindle fiber per chromosome. And here we have anaphase 1, where the homologous chromosomes separate to opposite poles. This is known as segregation, which is diploid to haploid. Individual chromatids in the replicated chromosomes do not separate yet. And here we have telophase 1. This is where chromosomes arrive at opposite poles, and two haploid cells form. Chromosomes may become chromatin and enter into interphase 2 in some cells. Now finally we come to cytokinesis. This is where haploid cells now enter into meiosis 2. Okay, so now we're into meiosis 2. This is when things start getting a little complicated. Here we have prophase 2. Here we have new spindle fibers forming from the centrioles, and the nuclear membranes start disappearing, as you would expect, and the chromatin condenses into chromosomes, as usual. Now we're on to metaphase 2. Here the chromosomes line up at the metaphase plate, and the chromosomes are made of two chromatids at this point. So here we have anaphase 2. This is where the centromeres split and individual chromosomes move to opposite poles. Okay, so this is where things start getting a bit tricky. This is telophase 2, and each of these has split off from a different part of anaphase 2. Here the chromosomes arrive at the poles, and the cytoplasm starts to pinch off, so it starts splitting apart, and you can just see that it's separated. Okay, and so now we're at the end of meiosis, and we have here the cytokinesis. So there's 4 haploid cells being produced. Each one is produced from half of the, from telophase. So that's where that comes from. Okay, so that was my really, really crappy crash course, if you will, of mitosis and meiosis, both meiosis 1 and meiosis 2. Hope you like. Please don't hate me for not knowing anything.